So we begin this semester with a tour through the circulatory system. Over the next 3 chapters, we'll cover blood itself, the heart, and then blood vessels and circulation. But in this chapter concerning blood, we'll look at the different types of erythrocytes, blood types, the different types of leukocytes, and then the platelets and how they're involved in homeostasis and the control of bleeding. The circulatory system consists of the heart, blood vessels, and blood. The cardiovascular system refers only to the heart and blood vessels. Hematology is the study of blood. The circulatory system has many different functions, the first of which we'll consider is transport. Oxygen, carbon dioxide, nutrients, wastes, hormones, and stem cells all circulate freely in the blood. The circulatory system also has function in protection. It's involved in inflammation, limiting the spread of infection by barricading off different areas, destroying microorganisms and cancer cells, as well as neutralizing toxins and initiating the process of clotting. The circulatory system is also involved in regulation, particularly fluid balance. It's involved in stabilizing the pH of the extracellular fluid in the tissues, and it's involved in temperature control. The average adult has 4 to 6 liters of blood. Blood is a liquid connective tissue. It consists of cells and extracellular matrix. Plasma is the matrix of the blood. It's a clear, light yellow fluid. It also contains formed elements. The formed elements are the blood cells and cell fragments, so red blood cells, white blood cells, and platelets, which are just fragments of cells. There are 7 different kinds of formed elements that we'll explore. The erythrocytes are the red blood cells, or we'll refer to them as RBCs. The platelets are formed from fragments of special cells in bone marrow. Leukocytes are the white blood cells. There are 5 types of leukocytes that are divided into 2 categories. The first category are the granulocytes. Those are sites or cells with granules. They include the neutrophils, the eosinophils, and the basophils. Then there are some agranulocytes. Those are cells without granules. Those are the lymphocytes and monocytes. We'll be exploring the roles of these in much detail as we progress through the next few chapters. The ratio of formed elements to plasma can be seen by taking a sample of the blood in a tube and spinning it for a few minutes in a centrifuge. This is called hematocrit measurement. The erythrocytes are the heaviest and they settle first, and they should be about 37 to 52% of the blood volume. The white blood cells and platelets are only about 1% of the total volume. They make up a narrow cream or buff-colored zone called the buffy coat, just above the red blood cells. The plasma is the remainder of the volume, so that's about 47 to 63%. It's a complex mixture of water, proteins, nutrients, electrolytes, nitrogenous wastes, hormones, and gases. When the blood clots and solids are removed, the remaining fluid is the blood serum. Serum is essentially identical to plasma, except for the absence of the clotting protein fibrinogen. There are 3 major categories of proteins that we find in plasma. Albumins are the smallest, but most abundant. Albumin serves to transport various solutes and buffer the pH of plasma. It also makes a major contribution to 2 physical properties of the blood, its viscosity and its osmolarity. It also has an influence on blood pressure, the rate of blood flow, as well as fluid balance. Globulins are antibodies, and they're divided into 3 subclasses. From smallest to largest, they're called alpha, beta, and gamma globulins. Globulins play various roles in solute transport, as well as clotting and immune functions. Fibrinogen is a soluble precursor to fibrin, which is a sticky protein that's involved in the clotting of blood. Plasma proteins are formed by the liver at about a rate of 4 grams of plasma protein per hour. It contributes all of the major proteins to the blood except for the gamma globulins. And the gamma globulins come from plasma cells in the connective tissue that are descendants from white blood cells that are called B lymphocytes. So protein is certainly the most abundant plasma solute. However, there are several other important components to consider. First of all, there are nitrogenous compounds. Nitrogenous compounds are toxic end products of catabolism. The most abundant is urea. Normally, the urea is removed by the kidneys. Other nitrogenous compounds come from free amino acids that are released from dietary protein or tissue breakdown. The plasma also transports nutrients absorbed by the digestive tract. These include glucose, vitamins, fats, cholesterol, phospholipids, and minerals. The blood also transports dissolved oxygen, carbon dioxide, and nitrogen, as well as electrolytes. Sodium ions constitute about 90% of the plasma cations. It's more important than any other solute for the osmolarity of blood. As such, it has a major influence on blood volume and pressure, so much that people with high blood pressure are asked to moderate their sodium intake in order to decrease the sodium and decrease. So now we've covered all the components of blood. Take out a pen and paper, close your books, and see what you can do to do about a minute of recall. What are the components of blood? We have the plasma, and we have the formed elements. What's in the plasma? And what are the formed elements? Do your best to recall as much as possible without looking at your notes or the lecture. After you've challenged your mind as much as possible, then open up your text and fill in the details. You'll make your brain hungry for knowledge. So there are a couple of properties of blood that we need to consider. First of all is viscosity. Viscosity is the resistance to flow, which results from cohesions of its particles. Really, it's how sticky the blood is. Whole blood is about 4 to 5 times as viscous as water, and plasma is only 2 times as viscous. So it's important to have the right amount of plasma relative to formed elements in order to have proper circulatory function. If someone has a red blood cell or protein deficiency, it would reduce the viscosity of blood and cause the blood to flow too easily, whereas an excess would cause the blood to flow much too sluggishly. Osmolarity, on the other hand, is highly connected to the idea of viscosity. It has to do with the total molarity of the dissolved particles that cannot pass through the blood vessel wall. If the osmolarity is too high, the blood is going to absorb too much water and increase the blood pressure. If it's too low, then we'll have too much water staying in the tissue and blood pressure drops and edema occurs. The optimum osmolarity is going to be achieved when the body regulates sodium ions, proteins, and red blood cells. So take a moment here and differentiate between viscosity and osmolarity to yourself. Yes, it has both have to do with thickness of the blood. However, one is how much cohesion there is between particles, how sticky it is, and the other simply is about the osmotic draw of fluids from the tissues and into the blood vessels. The components of the plasma, the fluid portion, needs to move out through the blood vessels and into the tissue and back from the tissue into the blood vessels. And only the proper osmolarity of the blood will allow passage to maintain things in homeostasis. There are several conditions that lead to hyperproteinemia, which is a deficiency of plasma blood protein. It can occur from extreme starvation or dietary protein deficiency, sometimes from liver diseases or kidney diseases that interfere with protein synthesis or result in protein loss through the urine. Severe burns can sometimes result in hypoproteinemia, loss of proteins through the body surfaces. When the protein content of the blood drops, so does its osmolarity. Then, when the bloodstream has less osmolarity, then it loses more fluid to the surrounding tissues than can be reabsorbed by osmosis. And the tissues will become edematous. And eventually you'll see a pool of fluid accumulating in the abdominal cavity, which is a condition called ascites. Kwashiorkor is an extreme version of this. It's seen in children who suffer from protein deficiencies in their diet. Their arms and legs are emaciated from a lack of muscle, and the skin is shiny and tight with edema, and the abdomen's usually swollen by ascites. We see this frequently in third world children who are no longer breastfeeding and now have a low protein diet of rice. So the process of hemopoiesis is the production of blood, especially its formed elements. The adult produces about 400 billion platelets, 200 billion red blood cells, and 10 billion white blood cells every day. Hemopoietic tissues are those that produce blood cells. The initial hemopoietic tissue is the yolk sac. It produces stem cells for the first blood cells. Those will eventually migrate into the embryo proper and colonize the fetal bone marrow, the liver, the spleen, and the thymus. Now, the liver produces blood cells during development and ceases doing that at birth. The spleen will remain involved only in lymphocyte production in adulthood. The red bone marrow produces all 7 formed elements throughout life. There are pluripotent stem cells. They're named this because they have the potential to develop into multiple different mature cell types. They multiply at a relatively slow rate and thus maintain only a small population in the bone marrow. Some of them will go on to differentiate into a variety of more specialized cells that are called colony-forming units. The colony-forming units will eventually become one particular class of formed elements. We'll describe this process later on in the chapter. Myeloid hemopoiesis is blood formation in the bone marrow, whereas lymphoid hemopoiesis is formation in the lymphatic organs. So that brings us to the end of the introduction to this chapter. In the next section, we'll move on to specific details about erythrocytes.